transport to me, when you think about the way that I describe it, it's almost kind of intuitive. So there's two different kinds of transport, and these are just ways that you get things in and out of the cell. Okay, so you bring things into the cell. Cells need to get rid of things that might be in the cell. There's two main ways that they do this. Passive transport is really what we're going to focus on, because passive transport is how you move gases like oxygen and carbon dioxide. They move by passive transport. Nutrients, waste molecules, these all move by passive transport. So the cell uses that cell membrane to maintain the barrier, right? So remember that that's the phospholipid, the bilayer. And I told you that that makes a great barrier to things that are polar or ionic. So if you have like sodium outside, it can't pass through because those little tails that are making in the middle of that bilayer, they're nonpolar, they're hydrophobic. So they are going to repel anything that's hydrophilic, anything ionic or polar. But the cell has to be able to move things in and out. It has to be able to communicate with other cells. It has to be able to get nutrients, get rid of waste molecules. And so it does that, which is why they call the membrane of a cell semi-permeable. So it's not completely permeable because then things could move freely in and out. Instead, it's semi-permeable, and that means that some molecules can pass, but others cannot. Passive transport is always going to move molecules from high concentration to low concentration. So this is sort of spreading things around. So if I have a lot of molecules on one side of the membrane, passive transport means they're going to want to move to the other side of the membrane so that they're equal, right? Having a lot in any one place, remember chaos, disorder. Disorder is like spreading it around. That's the natural tendency of things to do. So passive transport is always just going to move things from high to low. And so that's like one of the criteria. The other thing is that there's no energy required. So what I mean is it happens automatically. So osmosis, diffusion, facilitated diffusion, all of these are examples of passive transport. And that's just sort of the thing to remember about passive transport, is that we're always talking about moving things from where there's a lot to where there's a little, and we're never going to require the cell to use energy to make it happen. It just happens on its own. Although, if a molecule is polar or ionic, if we're talking about salts or we're talking about nutrients like sugar, which is polar, if we're talking about molecules that can't pass through the bilayer, they're going to have a channel. As long as there's a channel, then the channel will allow those molecules to go from high concentration to low concentration. So the first example of this is what happens in osmosis. So osmosis is the first example of passive transport. So this is the first type of passive transport. So you remember that we are roughly 2/3 water. Our bodies are mostly water. You have water inside of the cell, water outside of the cell. So when we draw that cell, there's concentration. Inside of the cell, you have salts. Inside of the cell, you have nutrients. Inside of the cell, there's hormones, proteins, other molecules. But outside of the cell is that way too. So you have all of these solutes. But remember that the solutes are dissolved in water. So that means not only do we have the sugars, the proteins, the vitamins, we've also got water dissolving everything. There are channels in the cell membrane that just let water move. So water can freely move in or out of the cell through little water channels. And water channels only allow water to pass through. So water channels don't let glucose to go to. It doesn't let sodium ions to slide in. It actually only allows these water molecules to move. Because oftentimes the salts and the nutrients, the wastes, they can't move. They don't have the freedom to move through. They have gates on their channels, so they don't have the ability to freely dissolve through those layers. So water is going to move to try and balance that. When they are balanced, that is a term that is referred to as isotonic. So if the amount of salts and nutrients, waste, dissolved molecules in and out of the cell is equal, then they say it's isotonic. The word iso means equal, and tonic really refers to the solute. So if the amount of salt, if the amount of sugar, if the amount of nutrients, gases, wastes are equal inside and outside of the cell, then they say that's isotonic. If you give a patient an IV, you're going to give a patient an IV that's got an isotonic condition. So it's going to have the normal amount of glucose present, the normal amount of sodium, the normal amount of chloride ions. So they're trying to make sure that it's balanced, because otherwise you can have shifts in water. If there is more salt outside of the cells, then the water is going to try and balance that by moving out of the cell. So here's the first example. If somebody ingests large quantities of water, that water gets absorbed. So water can get absorbed through the lining in your mouth. It gets absorbed through the stomach. It really gets absorbed quickly. It doesn't have to get digested or anything. It just gets absorbed through the lining of the GI tract. So if this water comes in, then it is going to dilute the blood. So if you think of your cell and then the blood outside, if there's more water outside, because they drank all this water, so if the volume and the amount of water outside of the cell goes up, that means that relatively there's less water inside compared to the outside. So what did we say that movement always happens from? High to low. So where is there at high concentration of water? Outside of the cell, less inside. What will happen to the cell? So see how there's more water here? So you could think this is high concentration of water now in the blood, low concentration, because this person just drank a lot of water. So you drink a whole lot of water, so you just sit and drink like an entire liter of water in 5 minutes, okay? So that means all that water then is going to get absorbed through the lining in the GI tract, pretty much like your mouth and stomach, get absorbed quickly, and that can dilute the blood, which means now there's more water in the blood, in the fluid part of the blood, than there is inside of the cells. So water is going to move from high to low. So water will go from areas where it's at high concentration to areas where it's at low concentration. So in theory, what will begin to happen to that cell? It's going to start to swell. So that cell will begin to swell. This, when they talk about the solution, they're actually referring to what happens to the blood. They say the blood becomes hypotonic. Hypo means what? Hyper means high. Hypo means low. So this becomes low solute. So do you see if there's more water than solutes, that the solute level goes down outside of the cell? And it's always outside of whatever you're looking at. When we did the potatoes in lab, remember that we put the potatoes in distilled water. And the potato actually gained mass. It got really hard. It wasn't bendy. That's because it was hypotonic. So if a patient has too much water in the blood, that can cause cells to begin to swell. So there is a good way to get rid of this excess water. What organ is responsible for helping to remove it? How do you get rid of? Kidneys. So normally, if you drink a lot of water, your kidneys will kick in because the kidneys are like, oh, there's too much water, not enough salt in the blood now. And so the kidneys start filtering that water furiously. And so you drink your liter of water, 20 minutes later, what are you doing? Right? I gotta go to the bathroom. Back and forth. Okay? Until it levels back off. So your kidneys are really good at trying to kind of like fine tune this, but you can have some swelling. The only time you really worry about this is if you have a patient that's dehydrated. So if you have a patient that's dehydrated, that means that they have very low water content in their blood. If you hydrate them really quickly, then you can have this shifting. So their blood has very low water. If you add water too quickly, then that can cause those cells to actually swell. Worst case scenario, that can cause fluid to begin to move into areas surrounding brain tissue. That can cause some hypertension. Cerebral edema can actually occur. So that's worst case. So like if you've ever been really sick, like vomiting, fluid, the whole 9 yards, when you start feeling a little better, what does your mother always tell you to do? Take small sips. Like don't drink all that at one time. Drink a little bit and wait. It's not an old wives' tale. It is because if you're in dehydrated condition, then the amount of water in the blood is actually lower than it should be. So adding a lot of water in could cause this shift of water into cells. So that's really the only time you're going to deal with a patient that might have hypotonic issues, like a puppy being brought in that's severely dehydrated. Okay? You want to hydrate them, but you don't want to do it like super fast because of fluid shifts that can actually occur. Opposite though, this is what happens in dehydration. So again, if you think of like a cell, if you lose water, you have a fever, you have diarrhea, you have vomiting. So all of this is going to be consistently like losing fluid. If we have a low water concentration outside because of illness, that means that relatively inside of the cell, the concentration is what? So we've lost water due to fever, vomiting, diarrhea. All of those can cause fluid loss, being sick, having a really bad case of the flu. So comparatively, inside of the cell, you have what? More water. So if you lose water from outside the cell because of fluid loss, now which way is water going to go? Where's water at higher concentration? Inside the cell. So that means water is going to start to move out. So water starts to move out. Remember, this is the potato that you put in the high salt solution, the one that got kind of brownish and really bendy. Like you could take and kind of squish it, was real spongy feeling. Same thing can happen to a cell. This is consistently happens if you have in dehydration. So the cell can actually begin to kind of shrink. They call that crenation. Under this, because there's not much water in the fluid outside of the cell, they say that the water level's low, which means the salts are high. So this is a hypertonic condition because it's a higher solute because of that loss of water. So somebody that's dehydrated, how do they look? Okay, pale. You may actually notice if they're truly dehydrated, you will notice their eye sockets, like their eyes actually look almost sunken. Have you ever had a patient where you took and pulled up their skin and it stays like that? Okay, I've had that with puppies. Okay, that's always the big worry. You're like, uh-oh, what's causing this? Okay, so it'll actually stay tented. And that is because of fluid loss in subcutaneous tissues. Because as you begin to lose water from the blood, you begin to pull water out of tissues. So the skin will even look kind of saggy. Like their face looks kind of saggy. It doesn't have a lot of the suppleness left. And that's all because of that water loss due to dehydration. So again, with this, you don't want to give them a whole lot of water really fast. You just want to give them isotonic saline, isotonic fluids to try and bring this back slow so that the cells kind of return to normal, fluids inside of the body return to normal, because you really don't want it just to like all end up like shifting too quickly. But dehydration, that can actually end up impairing the nervous system. So like the person may seem kind of disoriented, almost foggy acting, like difficult to answer. And it's because loss of fluid in the brain starts to lose fluid around it as well. So you can lose fluid surrounding the brain. So you have like this sort of cerebral shrinking that occurs. That's hypertonic. So think of hypertonic as when there's not enough water, too much salt outside of the cell, and it causes them to shrink up. Hypotonic is if you have too much water, not enough salt, and that can cause cells to swell. So I kind of like this picture because this picture sort of shows you the same conditions. So in isotonic, remember this means equal. So this is when the solute and water in and out of the cell are the same. So if we were going to be talking about salt solutions, 0.9% NaCl, that is called physiological saline. Physiological means that's normal body conditions for humans. So physiological saline, if they're equal, inside and outside, then the cell doesn't change shape. Water in and water out would be equal. So if they're exactly the same inside of the cell, outside of the cell, that's isotonic. So giving physiological saline would be an isotonic solution. Giving them what they call D5W, that's dextrose, which is glucose, 5% in water. So that'd be 5 grams of glucose per 100 milliliters of solution. That is isotonic glucose. So that'll help to get your blood sugar right to 80. Okay, so that's kind of like, remember I told you 60 to 110 is normal. So if we can get your blood sugar up to 80, then that's going to be like right in the middle of that balanced amount. But hypotonic, this is when you have less salt, more water outside of the cell, and see how the cell can swell. So these are just showing like electron microscope pictures of red blood cells. So this is when outside of the cell, you have very low sodium, higher water relatively to the inside. Because different solute concentrations, so we'll say salt or sodium chloride. So if there's a difference in the concentration, water's always going to try to move towards area where there's more solutes, because the goal is to try and equal them, right? So if you have an area where there's a lot of salt, water is going to move towards that area of higher salt to try and balance them out. That's why the cell swells, because the water is going to move from high to low. And the exact opposite occurs when someone's dehydrated, because hypertonic conditions is when you have a relatively high concentration of salt outside of the cell and very low water because of that loss of fluid. Going to cause that shift, and water will move from the cell out into the fluid and cause it to shrink. So you can see that red blood cell, they kind of start looking like little raisins. They look like somebody has deflated them. So just remember in these, like this is the potato in water. This one's the potato in salt water. And because different potatoes seem to have different water concentrations, I really have a hard time making a solution that's actually isotonic in all conditions. Red potatoes, russet potatoes, they all seem to have a little bit different solute. So I'm like, I just give up. I can't make them isotonic all the time. Go ahead.
The two arrows.
Mm-hmm. Okay. So if, and when we talk about hypotonic, okay, hypo is low. Tonic is solute. So see how, and it's always referring to outside, right? So see how it's low salt outside? That's hypotonic. See how the third one is high salt outside? That's hypertonic. So it's movement of water, but the name refers to the solute, which sometimes seems counterintuitive, right? You would think that it would, like, the name would refer to the water, but the name's talking about the solutes that are dissolved, but the water is what moves. The water moves to try and balance out the solute difference. So if you have a lot of salt out here, water goes to it. If you have more salt inside, water goes towards it. Okay, so that's like a common way that people talk about it. They're like, that water always follows the salt. The water is always going to try to go where there's more salt to try and equal their concentrations. Okay, so that is osmosis. So in this one, you can see, you tell me, when looking at this, do you see that this is the all of this here? There is the little yellow balls, little green balls. Do you see that there's less of them? So this would be like a low solute. Over here is high solute. A lot of red, a lot of green balls, a lot of yellow. So those would be the solute particles. So the kind of gray area, the space in between, all of this would be water. Well, you can't see green in that. This would be water. All over here, this, all that like blue space. So can you tell that like on the left, there's more water? There's more open kind of space. So there'd be more water to the left. And that's going to force that water to move to the right from where there's not a lot of solutes, but there's a high water concentration, over to the right where there's a lot of solutes. And the goal is to try and make them equal, to try and make the amount of water compared to salt the same. So this is this normal flow. And remember, this is passive. So you don't have, your cells don't have to do anything. This is automatically going to happen. Like the goal is always try to make them balance. So things don't go from high to low concentration to create another low to high concentration. They move until they have equal concentration. So water will move until the water concentrations on both sides are exactly the same. And at that time, then you would say that's isotonic. And when it's isotonic, nothing moves. It'll just equilibrate. It's sort of like at equilibrium. It's balanced. Okay, so we kind of talked about those. So then diffusion. Diffusion is the other passive transport. And this first one is really what I would call simple diffusion. Simple diffusion is the movement of solute from high to low concentration. And so can you see in this one says if you took a drop of green food coloring and put it in water and let it sit, it would spread out until it was uniform. The entire thing would look greenish, right? So we did this when we were doing the dialysis tubing. I just had you stir your tubing every once in a while to help speed the process up, right? And I told you, like, you could put sugar in your coffee and just leave it. Eventually, the sugar would actually diffuse and the entire thing would be sweet. It's just a little faster if you stir it. Just speeds up that process, but diffusion will happen whether you stir it or not. High concentration to low concentration, and it's the solute when you talk about diffusion. When you talk about osmosis, it's water or solvent, but for our case, it's always water. Passive diffusion or simple diffusion is a better term. This is when you have these small gas molecules, oxygen, nitrogen, carbon dioxide. They are nonpolar. So, nonpolar molecules, what do they do when they come to that phospholipid bilayer? So if you have oxygen, so oxygen leaves the blood and enters the tissues. In your tissues, your body cells need oxygen. So if we have, outside of the cell, lots and lots and lots of oxygen begins to build up. So here now we have a high oxygen concentration, and that's outside the cell. But inside the cell, there's none. So diffusion says oxygen wants to go where? Outside of the cell or inside? It's going to want to move in. That barrier is nonpolar, but so is oxygen. So oxygen can just slide straight through. There is no barrier for a nonpolar molecule, so it just moves straight through. So diffusion of gases, like what happens in your lungs, pulling oxygen from the air sacs in the lungs, pulling it into the blood, it moves just by simple diffusion. These molecules are not, there's no barrier of the cell membrane to them. They can just pass through that phospholipid bilayer because they're nonpolar. They're gases, so they either bind to hemoglobin and get carried or they dissolve in plasma to get carried, but they're able to move without any kind of energy, and they don't even have to have a channel. They can just slide straight through. So there's another type, and so really this is, you can tell a chemist wrote this. This is really what simple diffusion is. Just think of simple. Simple is what gases do. Oxygen and carbon dioxide, they just go from high concentration to low concentration. They just move through walls. They don't have a problem. Facilitated transport, though, this is how ionic and polar molecules get in or out of the cell. Doesn't require energy, but it may require a channel. So things like glucose, sodium ions, potassium ions, vitamins, the polar vitamins, they get to be high concentration outside of the cell. To be able to get into the cell, they have to use a channel. But it's still passive transport because it's going high concentration to low concentration. So just like water did in osmosis, things that can do diffusion are always going to move from high to low. So here's like their diagram, their image, and I think it's a pretty good one because see how it shows like simple diffusion, facilitated diffusion, the difference between them. So in this, the first one, see how the little red ball just goes right through? And it goes from high concentration to low concentration. So anything like carbon dioxide, oxygen, Those would be examples of things that can do this. That would probably be oxygen molecules because they want to diffuse in. Carbon dioxide diffuses out. But facilitated transport, this would be what glucose does. So glucose, when it's high concentration in the blood and there's insulin, that opens those glucose channels and that allows the glucose to move straight through into the cell. So then this last one, this is not passive transport. This is what they call active transport. How is active transport different? Yeah. So one requires energy. What's happening in active transport? See how it looks like it's going low to high? So you're taking that one blue ball and you're putting it where there's a whole bunch. This is creating order. That's never going to happen on its own. So it is going to move molecules from low concentration to where it's at high concentration, creating more order never happens on its own. But your cells need to do this because this is one of the easiest ways to be able to do jobs, to do the work that the cell needs to do. So for example, a nerve impulse. Nerve impulses can only be generated if you have an imbalance of sodium and potassium. So you actually have to make an imbalance. You don't want it to be balanced. You want to end up having a nerve cell that has more potassium inside of the cell, more sodium outside of the cell, because that's what gets exchanged in a nerve impulse. So reasons why you would have active transport. Nerve impulses require active transport. Muscle contraction requires active transport. In order for a muscle to contract, calcium has to be released into the cell to interact with the muscle fibers. So if you can't put calcium at a high concentration somewhere, you can't trigger that interaction. So active transport, I always think of it as like you use energy to do this, but it's important to do very important work that different cells have to do. It's not trying to maintain balance. It's not trying to make things equal like what the passive transports are. So now here's the application for this. So the application for this is dialysis. And in fact, dialysis is a process where both osmosis and diffusion help to balance the blood. So first off, and we're going to talk about it in terms of human, because I really don't know the number of nephrons in a dog, okay? So in terms of human, kidneys, the kidney's job is it filters the blood. The kidneys pull out all the waste. The kidneys pull out extra water and extra salts that you don't need. So like, you know, you go have like the Southern meal where you have like a lot of salt on your food. That means that your sodium levels are going to go up. That means your water levels are going to go up because the sodium, wherever salt is, water likes to go too. So you're all kind of edema, puffy legs, ankles, fingers type of thing from your dinner. But your kidneys are amazing because your kidneys recognize that. Your kidneys constantly look at the amount of water, the amount of salt that's in the blood. The kidneys go, way too much salt, so we have to start filtering this out. So it takes about 24 hours. But the kidneys will filter the excess sodium out of the blood. And so you'll notice that the edema kind of goes away over a 24-hour time. Right? So you might notice it straight away, but it takes a while to go back. That's the job of the kidneys. So the kidneys are going to remove waste and they're going to balance out small or water and salt. Ideally, all nutrients get reabsorbed. Okay? All the nutrients and a balanced amount of water and salt stay in the blood. Anything that's excess, so the wastes, excess salt and excess water, they don't get reabsorbed and they end up being excreted as urine. So you have these structures in your kidneys. They're called nephrons. These are microscopic filters, and you have 1 million of them in one kidney. So a nephron is just the little microscopic filter for the blood, and you have 1 million filters per kidney. So roughly you have 2 million nephrons. Those 2 million nephrons can actually filter your blood collectively about 35 times a day. So roughly it's not quite every 30 minutes, so it's maybe like every 40 minutes, your entire blood supply floats through your kidneys and filtration happens. It's that quick. Every 40 minutes, your entire blood supply passes through your kidneys. Excess water, wastes, and salt get pulled out, and that's what becomes urine. But if the kidneys get damaged, what was one of the things we said that kidney damage can occur? What's one disease? Diabetes, right? So diabetics, they have really thick blood when they have hyperglycemia. So those filters, they're like microscopically thin and really delicate. They do a good job, but they can't handle really thick blood. And so what happens is those nephrons get damaged. So yes, you start off with 2 million of them, but if you have less than 500,000, which means that's why you can donate a kidney, you really only need a half of a functional kidney. If you were born with excess all the time, as long as you have about 500,000 nephrons, you're good. But if it starts to go below 500,000, then they're struggling to keep up with filtering. So then those 500,000 are really trying to get rid of all the waste and balance the salt and water concentration. And sometimes they can't. When kidney numbers, filtration numbers drop below 100,000, so that's like you only have about 20% of what you absolutely have to have minimally, then they say that you're in kidney failure. So kidney failure means that there's not enough nephrons to remove the waste and balance the salt and water. So your blood now, waste molecules build up, salt and water concentrations get all out of whack. That's when you have to have what's called hemodialysis. So dialysis is a way to artificially filter the blood. And it's really just working on the premise of passive transport. Things are moving from high concentration to low concentration. So if you have high salt in the blood, it's going to diffuse into the dialysis fluid because there's little microscopic holes to allow that movement. If you have too much water inside of the blood, that excess water comes out. Salts, or not salts, but waste move from high concentration in the blood into the dialysis fluid to try and remove them. So here's the picture. So you know, like I've had this actually zoomed for me. No. It zoomed and I couldn't make it stop zooming the other day. I was like, really? So, no. So in this, let's see. No. So looking at this picture, the only way that I can make this happen is I can go like this and show it to you, but I have to hold it there. So if you look, can you see that this person actually has there's 2 needles? So there's one that's there and there's one that's there. Can you see that it kind of looks like there's a needle coming out this way and a needle coming out this way on their arm? There's one going this way and one going that way. Okay, so they're actually removing blood and putting blood back in. So once the blood goes, let me try one more time. Okay, so here, so I think you can see it a little bit. See the two? Okay, so there's one on either side. So this blood gets removed. So blood, and it's actually, it's got some pressure on it, so it pushes it out, but it's actually pumped to make sure it keeps moving. So the blood moves out of the tubing. Well, it doesn't come out of the tubing. The blood moves through the tubing and it goes into this dialysis machine. So this dialysis machine, the tubing kind of does this sort of coil all the way through. As it goes through, there are microscopic holes in this tubing. Surrounding the tubing is called dialysis solution, and a dialyzing or dialysis solution is isotonic. So that means that it's got the equal amount of salt and water and nutrients that your blood should have if your kidneys were functioning. So anything that is in excess in the blood is going to come out. Anything that is not enough in the blood will go in. So you can have fluids moving from high concentration to low concentration, high concentration to low concentration. So like salt might come out, but maybe like potassium moves in. Glucose might come out, or maybe glucose goes in. It really depends on the condition of your blood. So this dialyzing solution would be like the perfect blood. It'd be like perfectly isotonic. So you're like, well, wastes always come out because isotonic blood should have no waste molecules. So the waste moves out, and the way that they keep this refreshed is they're constantly pouring in dialysis solution and draining it away. So this solution is constantly being replaced so that it never builds up an imbalance. So now it's going to stay. Okay, so if you look at that, so just think like anything in excess in the blood comes out, anything the blood needs can go in. Waste molecules always come out because there shouldn't be waste in the blood. That's the job of the kidneys to filter it out. So it pulls it that way. Once it's done, now this freshly dialyzed blood, which is basically isotonic, gets put back in. So that goes in. So in the blood, I give you as this example. Here's a patient. If their sodium levels are like 160, normal sodium levels should be about 140. So if that dialysis tubing has those microscopic holes, which way will the sodium move? It's going to go from high concentration to low, so it'll come out of the blood and go into the fluid. So the excess sodium is going to go from where it's at high concentration to low concentration. So what about potassium? It's going to come out as well. This person's potassium is really high. Usually what they do is they take some of the patient's blood and do an analysis to see exactly what the conditions of their blood is, to see how out of whack. But can you see it should be 140, 1.25. The concentration in the dialysis fluid, this is the isotonic. This is what it should be. If you had normal functioning kidneys, your salts and electrolytes would be like this. But potassium, 4.2 goes to 1. Now what about calcium? It'll go the opposite direction. So you see in the fluid, it's 1.25. So maybe this patient's a little deficient in calcium. So we can actually help to move things that the patient needs into the blood. Bicarb. Which way will bicarbonate go? It's going to go into the blood, passing through. All of these are small enough to pass through the dialysis or the tubing holes. Magnesium. It's going to come out of the blood because it's too high. It's going to go into the fluid. Chloride. It's going to go in to try to balance. And then glucose. So maybe this patient's blood sugar is a little low. So normal blood sugar, normal blood glucose, 5 milligrams per deciliter of blood. So you'll have some of the glucose will move in. It's just going to go, all you do is look, whichever side has more, it's going to move the opposite direction. Is this all going to move out? No. So eventually, the sodium is going to end up being 140, right? So the 160 would move, but it's constantly replaced, so more would move, constantly replaced, and eventually they'll be isotonic. So eventually the sodium will be 140, potassium will end up being 1.0. So they'll end up matching. They'll have 1.25 calcium, so it'll end up matching what that isotonic condition of the dialysis fluid. Now the other one, there are 3 primary waste molecules that are in your blood. One is urea, the other one is uric acid, and the last one is called creatinine. These are the 3 waste molecules. Urea is a nitrogen waste. Uric acid is from the breakdown of like muscle metabolism and such. So again, it's another one that has some nitrogen, but can also be like there's some phosphorus in it as well. Creatinine's another one from muscle breakdown. So these waste molecules, normally your kidneys flush them constantly. So on the isotonic side, there will be no wastes. So that means all the wastes go which direction? They're all going to move out. And they're going to keep doing that, keep moving, keep moving. So by the time this patient is done dialysis, their waste concentrations are really low and their salt levels are balanced. When their salt levels balance, the water levels balance, right? Because remember the whole tonicity? If you have isotonic, that means equal solutes. So if you can get the solutes equal, water will balance itself. So has anybody ever had a family member do dialysis? All right, so when do they go? 3 times a week.
Every night.
Oh, so she does peritoneal? Yes. So she has peritoneal dialysis. It's the second kind of dialysis. In peritoneal dialysis, you literally have a port so that you can actually drain fluid. You weigh the fluid because the fluid with a lot of waste will weigh more. And that tells you how much fluid to put back in. And that fluid is actually in your abdominal pelvic cavity. So it sits in the area that surrounds your intestines, liver, stomach, and waste will diffuse out of those organs, out of the blood vessels surrounding the organs, and kind of like diffuse into that fluid. And then the next day you drain it off, you weigh it, and then that tells you how much more to put in. Huge infection risk. How oftentimes has she had peritonitis? Because I mean, literally you have a hole. And so if you are not like scrupulous about this, if you like, you can't touch it, like you don't want to touch the opening, you have to like go through like a whole cleaning series. It is really awkward. Just trying to do all of these things and not like hit it with your arm kind of thing. Anything touches it, you can introduce microbes, and that's literally introducing something directly into your body cavity.
It's only here? You can't put it anywhere else?
Well, yeah, they have them. I mean, it's usually on this area, like where the soft, mushy parts in the abdomen are. But it's like literally a hole. It's a port.
So what makes them have to do that too? Like, how does
So oftentimes, if there's not a dialysis center close by, or if they can do it themselves, then they let them try that. But there's always such a huge, I don't know people that do that a long time. Most people I know end up going to hemodialysis because a dialysis tech or a nurse is overseeing this. So they're sort of like, to get them started and making sure they monitor this. So you go to a kidney center, the dialysis center, 3 times a week. How long does it take? Yeah. Average about 4 hours. So so you go on Monday, Wednesday, Friday. So you show up Monday morning at 8 o'clock and you're there until noon. You show up Wednesday, you're there till noon. And so the challenge is think of your blood when the wastes and the salts and the water all start getting out of whack. So it makes you feel bad. So the day before they have to, like the night and the morning of dialysis, you feel pretty crummy. Because some things are high, some things are low, nothing's in this isotonic condition. So you're like this, you go to the dialysis center and in 4 hours, your blood goes, it like throws you into isotonic. Most people are so tired after that, the rest of the day they go home and sleep. So it's like literally 3 days out of the week, you don't do anything except dialysis because you spend 4 hours in dialysis and then it takes you another 4 hours just to recover from dialysis. So it's that type of.
Can a person have like a 9 to 5?
I know people have done this for 25 years. 25 years. Mm-hmm.
When they're on dialysis, you can't really travel or nothing like that.
No.
That can't even work.
Yeah. I mean, it is a huge challenge.
If you wanted to travel, you can't say, I'm going to
Yeah, it'd be like, oh.
and then go to a center.
I have friends that are like dialysis nurses, so they're the ones that monitor, and they're like the stories. So people are like, I gotta go have lunch. You gotta unhook me. She's like, you can't leave. No, I have to. Like, So I mean, it is something like literally, it's either Tuesday, Thursday, Saturday, Monday, Wednesday, Friday.
What if they skip
Every other day, and then they still end up missing because there's 7 days in a week, so you end up missing a day. So those are the roughest days. So like, if you have dialysis on Friday, now Saturday, Sunday, you don't go to dialysis till Monday, so Sunday is kind of rough because Sunday everything's getting even worse, like getting all out of whack. But if you didn't have this, these people wouldn't be alive. Their waste molecules would build up so much, it would cause damage and loss of function. Neurological function would be lost. Organ failure would result because your liver can't manage all of these really high levels. Yes. So the only problem is kidney transplants, average lifespan of a kidney transplant is 7 years. Yeah. People think, oh, I'll get a kidney and I'll be fine for the rest of my life. No. And it's because the kidney is such a kind of like complex organ. It is very difficult to match it exactly, right? So remember I was talking about how the surface of cells have those markers? Well, your kidney is not just one kind of cell. You've got blood cells in there. There's connective tissue cells, epithelial cells. So you have so many different kinds of cells, and they have to make sure they match 80% or better. And even with that, because there's so much blood flow through the kidney, odds are that your immune system is going to pick up on something that's not right. And as soon as that happens, then you start the immune response and you start the problems of rejection. They have to take immunosuppressants. So they were some of the people like during COVID that were very concerned. I have a friend who has a daughter that has a kidney transplant. And he was like, they didn't go anywhere. They didn't see anybody. They like went to the grocery store. They wore a mask in public. And it was not, they were vaccinated, but they were worried about giving something to her because she's now immunocompromised because she has to take medicine to try and block that kidney rejection. He had a biology friend in Wilson. He passed away after years and years and years between he did dialysis, then he got a kidney, and then after about 10 years it rejected, and he couldn't go back on dialysis after that. So, but this, I mean, there's no way that these people would be alive otherwise. So it does give, it does create life, but that ability to survive longer, but that is a huge, big challenge. And it's really all just osmosis and dialysis is the premise. So with him, his veins were like, he had like just knots because so when those go in because of the pressure and everything, those ports will scar over. So he just ended up having like knots all over his arms and legs and like literally ran out of available vessels to access.
Is that what they call the tissue?
Yeah. And it's just scar tissue, and it's because of the pressure and the amount of time that you end up being on this, so then when they take the needles out, now it closes up. Well, you always get a little scar tissue, like even just if they draw blood, you get a little bit, but they use really little needles. These are not really little needles. These are bigger needles to try and get this done. But it still ends up taking like 4 hours. So yeah, so that's kind of the application of it and what you will end up seeing. But if you're going to go into nursing, I'm sure that you'll see patients that have veins that are sort of junky, is what they kind of call them, junky veins, because of all of that goes through. All right, so that finishes the material that'll be on that take-home exam. So in this, make sure for you guys, it's only 7 and 8. just chapter 7 and 8, because we finished all of chapter 6. The day class didn't. They have some chapter 6 stuff. So you guys just have chapter 7 and 8. It's 6 pages. I mean, it's no longer than any other ones. There's multiple choice questions. There's like some fill in the blank kind of questions. Everything that is on there, though, we have actually covered. So you should be able to go like through your notes and find information and be able to answer them without any problem. So the only thing I really want to do is I just want to cover the background information for acids and bases. And we are going to kind of shorten it. Remember, I really did shorten chapter 8, believe it or not. So in acids and bases, we're going to talk about characteristics of acids and bases, neutralization for acids and bases, and then we're going to talk about pH. And then we're going to move on into chapter 9. Or chapter 10, chapter 10, which is because I really need to get through proteins. Okay, that's like a major nutrient, so I'm like, I have to get through that. Like, if nothing else, I have to make sure we get through that is an absolute. So acids, some of these characteristics, like, think of like a lemon. Lemons, they're acidic, they taste sour. Acids typically taste sour. They're really good at dissolving grease. And one other characteristic of an acid is that it actually reacts with metals and can generate hydrogen gas. So a lot of the bronze statues end up having to be like resurfaced every couple of decades. And like the Statue of Liberty just got done having a facelift. And so the rain that hits these statues is slightly acidic. It's not strongly acidic, but it's about a pH of 6, so it's a little less than neutral. So that little bit of acid over years and years and years actually begins to dissolve the metal. So they went up on the Statue of Liberty and like made her nose was like starting to flatten out. And that's all because of the acids that's in the rain. So Bronsted and Lowry, they were like, there's two guys. First, there was Arrhenius, and then these two guys. And they both looked at acids and bases and were trying to like describe, okay, what does an acid do? So Arrhenius, he really looked at it and said, acids, when you put them in water, they produce hydrogen ions, right? So if you take HCl and you put it in water, it ends up forming H+. That's the aqueous. Those H+, they call those hydrogen ions. And then the chloride ions are there too. So when we're talking about hydrochloric acid, sulfuric acid, nitric acid, all of those produce those hydrogen ions. So he was like, yeah, that's how we define them. But Bronsted and Lowry said, well, we're not talking about just what an acid does on its own. We're talking about what does an acid do when you put it with something else. And so it has that hydrogen ion, but it always tries to give the hydrogen ion up to something else. So they said, actually, when we take hydrochloric acid and we add it to water, it will give a hydrogen to the water and create H3O+. So they call that, they say it's a proton donor because it tries to get rid of its H+, or it causes another molecule to take on that hydrogen. So they're very similar, but there was sort of like a difference. Arrhenius was just looking at an acid all on its own. Bronsted and Lowry said, well, how does it interact with other things? So here you can see in this one. So you see there's the water molecule plus a hydrogen ion, and it forms what's called a hydronium ion. That's that H3O+. So it can actually make water have an extra proton. And that's just a characteristic of what an acid wants to do. Then they went back and they looked at bases. And they were like, okay, characteristics of bases. Bases, well, they taste pretty bitter. So like coffee has a lot of base in it. Spices, like if you ever take a bay leaf and suck on it, it's a little bitter. Parsley, like if you put parsley in your mouth, chew on it, it kind of leaves this like taste in the back of your throat. I always think of like those vitamins, those prenatal vitamins I had to take. See, when I took prenatal vitamins, they were not gummies. When I took prenatal vitamins, there were this brownish orange, humongous horse pill. And like you would take it, swallow it, and then you'd be like, and you would like taste it for hours and hours and hours. They were horrendous. Not my daughter. She's like, no, I have a little gummy. It tastes great. I'm like, you don't know what it's like. But if you think of things that leave like this taste in the back of your throat, a lot of medications do that as well. That's that bitter taste. You have more bitter receptors on the back of your tongue, whereas you have sour receptors on the sides. It also feels slippery. And I always think of the ocean. When you go swimming in the ocean, you know how your skin feels really slick? That's because the ocean actually it's slightly basic. It's about a pH of 8. So Arrhenius was like, all right, when I put a base in water, bases in water like sodium hydroxide, if I add water to it, it's going to release the hydroxide ion. So that one should be familiar because that's one of the polyatomics. They'll both be aqueous. So that was his definition. Acids make a hydrogen ion, the acids make H+. Bases make the hydroxide ion, the OH-. Bronsted and Lowry came back and said, well, what do bases do when you put them with stuff? And so they said, oh, bases are the ones that take on hydrogens. So a hydrogen gets released and the base actually accepts it. So they called them proton acceptors. So base acids are proton donors and bases are proton acceptors. So let's look at that a little closer so you can kind of, I think, see it a little better in these examples. So in this top one, hydrogen or hydrochloric acid plus water. They just called it hydrogen chloride so you wouldn't know it was an acid. So hydrochloric acid in water. So do you see that the HCl, and this is kind of one kind of like you had to look at when looking at oxidation and reduction. So you're sort of doing a comparison. Do you see that HCl, the chlorine, loses its hydrogen? So that is why they say it's a hydrogen donor. I always think it loses it. So the acid on the reactant side loses the hydrogen on the product side. But the base, this water in this example, when I look at the water in this example, I can see that it goes from being just H2O to now it's H3O plus. So I can see that this gains the hydrogen ion in the reaction. That's why it's a base. Looking at the bottom one, if I take ammonia, which is NH3, and I add water to it, it can react and form an ammonium ion and a hydroxide ion. So what would you say actually happens here? NH3 to NH4 plus. What happens? Gains a hydrogen. So if it gains a hydrogen, it is a base, right? Just like the one above. The water gained the hydrogen in the one above. So the water in the first example is a base. And this one, NH3 becomes NH4. It gains a hydrogen. So that makes it a base as well. And water is kind of interesting because in this example, what is water doing? Water goes from this to this. So it loses the hydrogen. So it goes from H2O to OH minus. So it loses a hydrogen. And that makes it an acid. So it's really just like this swapping of hydrogen ions. So it's not really a swapping of hydrogen. It's a swapping of hydrogen ions that occurs. So the one that gains the hydrogen ion, that's always going to be the base. The one that loses the hydrogen ion, that's always going to be the acid. So some of these you should see and go, oh yeah, we've used these in lab. Strong acids, sulfuric acid. Remember, I always put that in a little dropper bottle so you would only get a few drops. It was a common catalyst that we use in reactions. Hydrochloric acid, HCl, another one that we use, like we use that in the neutralization for the antacid lab. Those are common acids that we use all the time. The other one we used in the very first night, I don't usually use this very much, is nitric acid. It's just really strong. Those are strong acids. So strong acid means that it makes 100% ions. So strong acids, they are strong electrolytes. So notice in this, the air goes in one direction and ions are formed. So remember from the last chapter when talking about like forming an electrolyte? If it's a strong electrolyte, the arrow goes from left to right and you have ions that are produced. Same thing, strong acids and strong bases, they are strong electrolytes. The strong bases, we used this, sodium hydroxide, we used that when we made soap. So that was the base that we had to add in. We haven't really used the other ones very much. I just circled the ones that are pretty strong. The term corrosive refers to chemicals that create a lot of reactive chemicals. Chemicals that release a lot of reactive ions. Strong acids and strong bases are called corrosive because strong acids make a lot of hydrogen ions. Strong bases make a lot of hydroxide ions, and both of those react with body tissues. So that's why, like when we were making soap, I was like, well, be careful, don't get any of this on you. So in that first lab, working with the egg white and the cotton squares, be careful, don't get any of these ones on you. Just make sure you get small amounts of each, because all of those would end up reacting with body tissues, breaking those body tissues down because of these very reactive ions. But there's also some examples of acids that don't form a lot of ions. And so in fact, they should have, they didn't put it in, but it should look like that. So this, you can have a weak acid or a weak base where the arrow goes both directions. "95% of these molecules don't form ions, just a small number do." So remember, that was the one that when we did the light bulb test, it was barely glowed. So those are the weak acids. So you can have weak acids like vinegar. You eat vinegar. You wouldn't eat hydrochloric acid, but you eat vinegar because it doesn't make a lot of ions. It still has an acidic characteristic, but you don't end up with a lot of ions, so it's less corrosive than a strong acid. So weak acids and bases make fewer ions. So they're less corrosive, less reactive with body tissues. So a lot of the things that you eat, like in tomatoes, coffee, soda, like carbonic acid is in soda, it's a weak acid. So it doesn't make as many ions, it's less corrosive, less damaging to body tissues. Vinegar. So then the last thing. So if we take in an acid and a base, and we actually did this, we talked about this in class. So if you take an acid, like HCl, it's a strong acid. And then you take NaOH, which is a strong base. If you do a double displacement reaction, the sodium will combine with what? The sodium of the base combines with what part of the acid? So the Cl. So that makes what? NaCl is salt. And then what's left, the OH of the base and the H of the water, of the acid, combine and form the H2O. So it starts with two things that make a lot of ions, and you end up with two things that are extremely stable. Table salt. It just makes salt water. That's why they called it a neutralization, because it makes the acid and the base equal something that is neutral and non-reactive. So there is a slide about antacids, which we did in lab. So antacids, when we use antacids, we actually use a weak base rather than, you don't want to eat lye, because that would cause a lot of damage to your mouth, to your esophagus, and even in your stomach before it ever neutralized stomach acid. But if you take a Tums, or if you take baking soda, or there's magnesium hydroxide, calcium carbonate examples here. So notice these bases react with HCl, which is stomach acid, and it makes a salt in water. So if you have excess acid, you use those antacids to help decrease the amount of stomach acid that's present in your stomach, and that helps to alleviate the indigestion. Sodium bicarbonate, that's baking soda. Calcium carbonate, that's in Tums. The milk of magnesia that we used last week doing the antacid lab, those are all good examples of bases that can be used to neutralize strong acids, and they're not themselves reactive because they're a weak base. They don't make a lot of ions. If they don't make a lot of ions, then they're not going to be as reactive to your tissues, but they'll still react with the acid. Okay, so I'm going to quit there. That's just sort of like the intro stuff for acids and bases. There's not a whole lot more that we're going to do with it in this chapter other than talk about like pH and buffers. So